cytoplasm really had this property. So over the years, people tried to purify it, and some very good biochemists tried to purify it. I mean, John Gerhardt was really the inventor of allostery, or one of the inventors of allostery, and Mark Kirschner is extremely good. They knew how to purify things, how to work on enzymes. They failed, but what they didn't fail to do is to chart the comings and goings of this mysterious substance, this substance which catalyzes entry into mitosis. And what they found was that it went up at first meiosis, then disappeared between meiosis 1 and 2, went up and stayed up, and we'll talk about that shortly. Then fertilization caused it to go away, and then it came back for the rapid cell divisions that occur after fertilization of the frog egg. And that's fine, but then you say, well, you know, who cares about how frog reproduction? We don't really. So some people in Japan looked at starfish, which I've shown you in the movie. That was starfish. And they found that just when the nuclei were breaking down in those starfish oocytes, you could detect MPF. And you could detect, amazingly, starfish MPF by injecting it into frog eggs. Gosh. And I think even frog MPF worked in starfish. So this stuff is sort of really transcends even phylum boundaries. So that made it more important. And then people actually discovered that dividing human cells contained MPF, and even yeast contained MPF. So, you know, it became very pressing. This appeared to be the catalyst for cell division, and people wondered what the hell it was. And because it couldn't be purified, they couldn't find out. So this is, in a very abbreviated manner, how I came on the scene, and I wanted to study these things, which are fertilized sea urchin eggs, simply because when sea urchin eggs are fertilized, I wasn't at all worried about switches, except one, which is how they activate protein synthesis. Protein synthesis in an unactivated, unfertilized sea urchin egg occurs at a very, very low level, and what happens when the sperm comes along, about 5 minutes later, there's a tremendous activation of the rate of protein synthesis. And I, at the time, was an expert on the control of protein synthesis. Actually, mostly in rabbit reticulocyte lysates, but I just thought it would be fun to look at these cells, because whereas I was used to studying a situation where protein synthesis turned off, in a sea urchin egg, protein synthesis is turning on. It seemed a little bit more positive somehow. Now, as you've seen in this movie, let me just run it one more time, what I hadn't realized was how, so this is about 1 hour after fertilization and every 30 minutes, and look how synchronous the divisions in this culture are. Amazing, you know. Quite extraordinary. And the first 3 are more or less, if you do it right, are more or less occur exactly. You could almost set your watch by it. And by doing a very simple experiment, just labeling these eggs and taking timed samples, what I discovered was that 1 or 2, depending on the species, of the newly synthesized proteins, whose synthesis was turned on by the arrival of the sperm, had this amazing property that they just disappeared. And then they came back again, and then they disappeared. And they disappeared every time the cells divided. And if you stopped the cells dividing by adding microtubule inhibitors, then the thing didn't disappear, and so on. Did a lot of phenomena. So people ask, you know, so why did you win the Nobel Prize? And it's sort of a little bit embarrassing to say, well, I won it for finding something that disappeared. Incidentally, my wife calls me, she says, I'm the People's Laureate, because if I can win a Nobel Prize, anybody can. And it's kind of true. I mean, you know, this wasn't exactly the world's most technically difficult experiment. In fact, it was an experiment. I mean, if anything, yeah, I'm pretty simple-minded, as you'll see. So what was interesting about this, so it goes away, okay, fine, and it turns out that it's going away is very important. If it doesn't go away, you get stuck. So that is it, but we didn't discover that for a long, long time. But what's clear is the stuff accumulates steadily like this, whereas the switch occurs, and this is going to be a sort of theme, just bear this in mind. There is a, you know, you can't really say this must be MPF or a part of MPF, because it clearly isn't. I mean, MPF looks like that, and cyclin goes like that, so the two things don't correspond, and this bothered us a lot. Another thing that bothered us a lot was that early on, is it really true that, I mean, this is a radioactive band on a gel. Is it really true that can make enough stuff to catalyze cell division? I mean, you can't make very much stuff in half an hour or something. In fact, probably less. It was 20 minutes worth of synthesis. So we worried a lot about things like that. And, I mean, basically, the secret was to clone and sequence these things and then do lots more experiments. And it took quite a long time, especially as we weren't very good at cloning to begin with, because we'd never done it before. That's another lesson, actually, for young scientists. Just because because you can't do something doesn't mean that you shouldn't do it. It probably means even more that you should do it. And the trick is to go and learn from somebody who knows how to do it. And you'll find, at least I've always found, that actually if you approach people and say, you can do this, can I learn from you? People are amazingly generous and helpful, generally. I mean, there are some bastards out there who would... but the majority are not, actually, it turns out. And this is very comforting. Anyway, I immediately realized, given this result, I was terribly excited about it, and I immediately realized that I'd sort of stumbled into a completely different field than I was in. This was control of protein synthesis. Now I was in the control of the cell cycle. What was known? Answer, not very much. The number of people in this room will be about 3 times the number of people who worked on the cell cycle back in the early 1980s. Not because it was a boring problem, not because it was an unimportant problem, but just nobody could see quite how you study it. And the great pioneer here was really Lee Hartwell, who stumbled on a way to study it by using yeast genetics. And he focused on a gene called CDC28, which seemed to him to be an important controlling gene. It wasn't something involved in DNA synthesis, probably because it seemed to control lots of biochemically rather different processes. And then along came Paul Nurse, who followed in Lee's footsteps. He studied a different yeast, and if you make CDC mutants of this yeast, they just can't divide, so they get longer and longer. Here's the wild type. It divides at this length. This is a mutant, a temperature-sensitive mutant. And what Paul did by accident was to isolate this strain, the Wee1 strain, which divides at a much smaller than usual. So he realized that the cell cycle has been accelerated. And he then went on to isolate different strains of yeast which had the same phenotype. And try as he might, the first 49 strains that he went through all turned out to be alleles of Wee1. And I think it was the 50th or the 51st strain that he examined turned out to be an allele of a gene that he already knew about called CDC2. This is, in fact, the second one he'd isolated. So this gene becomes immediately very interesting because you can mutate it to inactive, in which case the yeast don't divide at all, and you can mutate it to hyperactive, in which case the cell cycle is accelerated. And so it must be a regulatory gene. And then it turned out that actually it was the same protein and interchangeable with CDC28, so it controls not 1 but 2 cell cycle transitions. And when it was sequenced, it turned out to encode a protein kinase, and then when it was found in humans too, people realized that studying yeast in a cancer institute was actually more justified than some people had thought. I mean, they actually criticized the guy who hired Nurse because, we already understand cancer so well, we should be finding cures for cancer, not studying yeast. Well, Paul shut them up very radically because they realized they didn't understand how cell division was controlled. And surely that must be a good thing to do. So it turned out that my protein, cyclin, was actually an activating subunit of CDC2, CDC28. And the way the thing works is that there's a rather complicated control, which we have to come back to later, of CDC2. It's controlled by an inhibitory protein kinase, that's the Wee1 gene, which is opposed or reversed by a protein phosphatase that takes that phosphate off. And the two of them together make MPF. So it works like this. The water